This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1111, recorded on May 3rd, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and Alan. I will just include Alan, even though he hasn't been introduced yet. I just got back from a 3-day fishing trip in the Catskills, so that's why my face looks a little red. And my hands, I can't tell you how sunburned they are. Well, I can show you. That's from holding a fly rod in one position and having the sun bake you like you were some kind of a gourmet dish for the gods. But I had a great time. Today, the temperature is about, I would estimate, I haven't checked it, but it feels like in the high 40s or low 50s. It's a little bit windy. It's typical springtime weather, but it's delightful. It's all the trees are blooming. All the leaves are unfolding. It's just my favorite time of the year.
It is here in Chelsea, 17 Celsius and partly cloudy. Right. I didn't see any sun this morning. Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and I apparently have the warmest weather. It's 67 Fahrenheit, 19 C.
Gee whiz.
Mostly cloudy. Inland. It's supposed to rain later on this weekend on Sunday, but just cloudy now.
I guess because you're inland, you have a higher temperature.
Yeah, we have different weather patterns than the coast.
So is episode 1111, that's an interesting number. We're just bantering about it.
If only we could have recorded it in November.
Exactly. That's right. Yeah. That's right.
You know,
on the 11th of November.
There was a year 1111, right?
There was a year 1111. We were not recording TWiV back then.
No, they weren't recording anything then.
No. But this is our first 4-digit. What do you say? All the same number. Is there a word for that?
Probably.
There might be. I wouldn't know it.
Then Dixon said, well, what about all the other TWiVs with just ones in them? So there's TWiV number 1, which was Dixon and I. That's West Nile's story.
Yeah, that's right.
That's easy. TWiV 11 was one of the ones where Alan was here.
Oh.
Actually, it was me, Alan, and Jeremy Luban.
Cool.
It was called Elite Controllers.
Oh, yeah.
Mosquitoes and winter vomiting. That's 11. TWiV 111. Live at Florida Gulf Coast University.
Oh, my.
That's when Rich and I went down there and had some guests, including, so it was the dengue control, Florida Keys Mosquito Control District.
I came in as a disembodied head on that episode.
You did.
Yeah.
And we had the pilot that flew these people around.
Well, and who flew the helicopters for the pest control operations.
A talking head.
Yeah, that's 111. And then 1111, well, we don't know what the title is yet.
We do not, but we probably can assume that Alan had the title.
Well, I don't know. We'll see. I have a few ideas.
If you like what we do on this program, This Week in Virology, and all the others that we produce here at Microbe TV, we would love to have your support to continue their production. It doesn't have to be a lot. It could be less than a cup of coffee a month. You know, coffee's expensive. Well, Vincent, I don't drink coffee. Okay, whatever your drink is, tea, you know, all the stuff with sugar in that you get at the fancy coffee places, which are not really coffee, but, you know, I go and I order a coffee and they're like, what? What's that?
Right.
We'd love your support. Just think of it as giving us a cup of coffee a month. Microbe.tv/contribute. You could set that up at Patreon or PayPal. A few meeting announcements. The pandemic preparedness meeting is November 11th through the 13th in Trieste, Italy, to provide an overview of known and emerging viruses and their potential epidemic risk. And we will put a link to that in the show notes. Also, the molecular determinants of zoonotic viruses and beyond. You getting a theme here about meetings? You seem to be focusing on emerging viruses, zoonotic viruses. Anyway, that one is in Freiburg, Germany, and that's going to be in 2025, March 1st is when it begins at the Institute of Virology in Freiburg, Germany. And don't forget Dixon's book, The New City, at despommier.com. In the news courtesy of Amy Rosenfeld, there's a Nature article called Chinese Virologist Who Was First to Share COVID-19 Genome. There's no COVID-19 genome. Doesn't exist, folks. Sleeps on street after lab shuts. The subheading,
COVID-19 does exist, but the virus is called SARS-CoV-2.
Yeah, no genome of COVID-19 exists. Yes.
Now, to be clear, his lab has not been shut down, and he's not being, as far as I can tell, he's not being like punished for anything. He just, there's a dispute about where his lab is being moved and how, and he's left outside the lab and not able to do his work, and so he's literally camping out on the street.
My goodness.
Why doesn't he go home?
I think he wants to make a point.
I see. So they didn't take away his home.
No. As far as I know, he has a home, but what's happened is that there's some snafu going on with a lab move and renovations, and this is very unfortunate.
They didn't take away his Eppendorfs, did they?
I don't know what the movers took.
That would be interesting. So if you may remember, at the beginning of 2020, he was among the first to sequence the genome of SARS-CoV-2 in China. And he is a collaborator of Eddie Holmes. And Holmes said, you need to release this sequence. People need to start working on it. He said, I can't. The Chinese government doesn't. They want to do it. And apparently Holmes was on the phone to Zhang just before he boarded a flight. Zhang was about to board a flight, or the door was about to shut, and Holmes said, you got to send it. So he emailed it from his phone, boom, and he got it. And then that was the sequence that was used to make the mRNA vaccines, essentially. And then China government got pissed at him. And, he's been persona non grata in China, I presume.
Yeah, I don't think so. From the article, I shouldn't say that he's not being targeted, but because I don't know what the exact situation is. But from the article, what I get is that this is some lab move has been handled poorly, and he's probably quite rightly upset about it, and is making his point.
I hope he's got a blanket.
Yeah, I hope that everything's okay.
He's not a young man.
Sleeping bag also.
Yeah.
Well, he would feel at home here in New York, that's for sure. All right, the next article is from the Jerusalem Post. Health Ministry announces measles case in Haifa. In the case of measles in Haifa, the patient had returned from abroad and left Bucharest on Wednesday, April 24th, and he was found to have measles upon arrival in Haifa, and also visited a urgent care clinic on the night of Friday, April 26th. So you can imagine that he or she may have transmitted the virus further at that health clinic.
Yeah.
Further, and if there are people who are not vaccinated against measles in Israel, this could be a problem.
Indeed.
The next one is a Reuters article. The UN says waterborne illnesses are spreading in Gaza due to heat and unsafe water. And so waterborne diseases are spreading. It's becoming hot there. People are getting much less water they need. There have been waterborne diseases due to lack of safe and clean water and the disruption of the sanitation system, which you can imagine is happening because everything's getting trashed, right?
This is a depressingly common problem in war zones, and that's what's going on. Yeah.
Since mid-October, following the assault on Gaza, WHO has recorded more than 345,000 cases of diarrhea, including 100,000 in kids under 5.
Those are the cases that came to the attention of the WHO. So this is
tip of an iceberg.
Yeah.
Gaza's only natural source of water is the coastal aquifer basin, which runs along the eastern Mediterranean coast. Its quality over the years has deteriorated, largely because it had been pumped out to meet the demands of Gaza's population more rapidly than it could be replaced by rainwater. This is what happens in in a war zone, as Alan said. It's highly unfortunate.
Yeah, other situations too, like when there's a flood or, you know, anytime the water supply is compromised.
Yep. We have a bioarchive preprint, which is interesting and maybe will become published one day, and maybe we'll cover it. Emergence and interstate spread of highly pathogenic avian influenza A H5N1 in dairy cattle. You know, someone said to me recently, if it's in another other than a bird, don't call it highly pathogenic. It's highly pathogenic in certain birds, but like in cows, it's not highly pathogenic. So highly pathogenic shouldn't be applied to it everywhere. So it's H5N1. So what is this?
Okay, so it's referred to, this is not like other viruses that have a name, controversial or not, but HPAI.
Yeah, it's a thing.
H5N1 doesn't tell you exactly which H5N1, right?
Yeah, there's HPAI and LPAI. Yeah.
Right.
Okay,
so this is the
enough.
specific HPAI. But yes, it is misleading to say highly pathogenic avian influenza A in dairy cattle because it's not highly pathogenic in dairy cattle.
But it is part of the name. You're right.
Yes.
You're right. This in North America, so viruses related to goose, Guangdong 2.3.4.4 BHA have infected wild birds, poultry, and mammals. And they did genomic and epidemiological investigations showing that a reassortment event in wild bird populations preceded a single wild bird to cattle transmission episode. And the movement of asymptomatic cattle has likely played a role in the spread of the virus within the U.S. dairy herd. Some molecular markers in virus populations were detected at low frequency that may lead to changes in transmission, efficiency, and phenotype after evolution in dairy cattle. Continued transmission in cattle increases risk for infection and subsequent spread to human populations.
So, Vincent, would you call that a lethal mutation?
Oh, boy. That's pretty good, Dixon.
Yes.
You should
No,
be on-
it may be an attenuating mutation.
Okay.
You could be on Colbert.
Yeah.
I was. on-
You were, yeah.
I know.
If it does become highly pathogenic in dairy cattle, that'll be the dark side of the moo.
That's right.
All right, now finally we have.
I'm sorry, I shouldn't joke. It is a serious issue that needs to be tracked, and we will.
Yes,
I think we're going to have
sure.
somebody talking. Don't we have a guest coming up?
We have a guest coming up who will talk about it.
Yeah.
An expert. Finally, from and Amy writes it because it's cool. Orangutan plays doctor, heals himself. This is in Science News, and this is very interesting. They find that an orangutan in Sumatra used a medicinal plant to heal a wound on its face.
That's right.
It's the first documented case of an ape using a plant with scientifically proven medicinal properties to treat a fresh wound. And, you know, it's not surprising because animals can learn what helps them and then they remember it, right?
That's right.
So some chimps chew bitter piths of the medicinal shrub Vernonia amygdalina to treat worm infections. Did you know that, Dixon?
Well, yes and no. They've looked into a lot of that mythology, and some of it has never borne fruit in terms of coming up with a new drug to treat worm infections with. So it's hard to know how much truth there is in that.
So this orangutan probably was involved in a fight.
And not in a flight.
In a fight. And they saw him eating leaves of Fibroaurea tinctoria.
But he also was applying it to the wound itself.
Yeah.
Yeah, it contains dozens of bioactive chemicals.
Amazing.
Yeah, so it's a medicinal plant that's apparently used traditionally and has been proven to have compounds in it that are relevant for this as an analgesic and for other purposes. And so this ape appears to have figured out that chewing on this particular plant helps kill the pain from the wounds, and then he was seen chewing some of it and putting the paste onto the wound,
Amazing.
which is right under his eye, and
it healed in a month.
Yeah,
but they didn't have a control.
They did not have a control. They did not.
I mean,
I presume he got the idea from watching a Nat Geo show.
Most likely, yes.
I mean, he was clearly putting it on there, which
I mean,
so he is self-aware, right?
Yeah.
he probably didn't look. He didn't probably see it because he has no mirror.
Doesn't have a mirror.
But he probably felt that it hurt, and he probably put his fingers up there. Oh, it's got quite a wound. You can see in the picture.
Yeah, he's got a real gash there.
And he's lucky he didn't lose his eye.
I don't know what the other guy looked like.
Yeah, you should see the other guy.
Right.
What an interesting animal, though, right?
Yeah.
Or, as they would say in the trade, you should see the other non-human primate.
Right.
It looks like his lip was banged up, too, and that healed.
Yeah.
Yeah, this is very cool. I'm very happy when animals, well, they've been doing it longer than we have, right?
You know, elephants seek out mineral deposits of various sorts, not just salt, but other minerals as well. And they remember where they are, of course. And animals are much smarter than we give them credit for. That's
well, they also know which plant to do, right? They don't
absolutely
just pick any plant. They pick the one that they have had
that's
experience.
Right. That's right.
Probably many, many years of experience. So.
And they know which ones not to eat.
Well, yeah.
That they learned years ago, right?
Just take one bite, forget about it. 3 weeks of diarrhea, I don't need that.
Don't eat that.
Okay, we have 2 papers for you today. The first is a Nature Communications article. MyADM is a nice word. We'll tell you what it is. It's myeloid association differentiation marker. It's a protein, okay? But it just has a name. Binds human parechovirus 1 and is essential for virus entry. This comes from Shao, Richards, Kim, Zengel, Ding, Greenberg, and Carrette. Jan Carrette is the PI here. And this comes from Stanford and the VA, Palo Alto Health System.
And Washington University.
Oh, yeah, WashU is on theirs too. So the reason I chose this, there are 2 reasons. There's always a reason. Folks,
There's always a reason.
Always a reason.
First, I wanted you to learn about parechoviruses. And second, it's a very clever approach to identifying a cell receptor for a virus. And I was always interested in these because my lab...
Coronaviruses.
These are parechoviruses. My laboratory, specifically Kathy Mendelson, one of my first graduate students. Did you overlap with Kathy?
No, she had just left when I entered the lab.
She identified the cellular receptor for poliovirus very early, in the late 80s, a time when we knew very few virus receptors. And when I talk about how this one was found, I'll tell you how we did it. It's just a totally different technology. Parechoviruses. So the parechovirus is a family that comprises like 40 genera, and one of those genera is enterovirus, which contains polioviruses and Coxsackie viruses and echoviruses. And the echoviruses used to be echoviruses. They just were echoviruses until Timo Hippia in Finland. He studied them. He said, these are really different. And then the genomic analysis showed, so these were discovered long before we were able to sequence genomes, showed that they were distinct members of the enterovirus genus. So now they're called parechovirus.
Why are they called echo? What's the word
enteric
echo
human cytopathogenic orphan viruses? Is that right?
Yeah, say it again because Dixon was
I muddle over you. Sorry.
Enteric cytopathogenic human orphan viruses.
Right.
Oh
not because they infect human orphans, but because the viruses were discovered and were not associated with any known disease.
That's right.
So they were thought to be completely asymptomatic. We now know that there are echoviruses that can cause serious problems, but a lot of them are asymptomatic.
So when Timo Hippia said these are different, he decided to call them parecho, right? So not that different, but what does the prefix par mean? P-A-R. Is there a meaning to that?
Don't know exactly.
Does it mean around?
Right. Para is close to or...
Par.
This is not ortho.
Right.
No. Anyway, so parechos, there are 19 genotypes, and the most prevalent are A1 and A3. They frequently infect young children and typically cause mild GI or respiratory symptoms. So you can have respiratory infections or GI infections. and they are being increasingly realized as to be causes of severe disease in young children, including neonatal sepsis, childhood meningitis, and encephalitis. So they're an emerging pathogen because we used to think they didn't do anything, which was just ignorance on our part.
So
Now-
they're not emerging, but our knowledge of them is.
Yeah. So
They've been
emerging
around for a very, very long time, but we're figuring out now that
Yeah.
they can be serious.
Emerging refers to not only a new virus to humans like SARS-CoV-2, but a virus that we've known about that has a new disease like Zika virus, right?
Yeah.
So, all right. So this paper looks for the receptor. This is one of the things you'd like to know for a virus to be able to understand how it causes disease and so forth. Maybe design some interventions, but that really doesn't work very well. And so they have a very clever way of looking for the receptor and then doing a series of experiments that show that this is a required receptor and where in the entry process it's working. So a little background on what we know about parechoviruses. Now, the genotype A1 binds to cellular membrane proteins called integrins. And there's a arginine glycine asp tripeptide on the capsid of the virus that binds to this. Arg-glyasp is a known ligand for integrins, and the virus capsid has it. But they don't know if you need something else besides the integrins. Like, do you need one or do you need multiple receptors? And plus, the genotype A3, they don't know what the receptor is. It doesn't bind to integrins, so they want to identify what the receptor is. So they do a CRISPR-Cas9 genetic screen. So they buy a library of lentiviruses that contain CRISPR-Cas9 and guide RNAs to target every known protein in the genome, the mRNA, the DNA encoding it.
Right.
So the idea is you infect a population of cells with these viruses. They're going to deliver CRISPR-Cas9 plus the guide RNA to each cell, and it's going to clip out a different gene because you have all kinds of guide RNAs in there. Now you grow up this population of cells, then you infect them with your virus and you see what's resistant to infection. So what survives? Because the virus is lytic, it will kill cells, and you ask what's left, right?
Yeah.
And then you take those and you can pull out the gene that has been disrupted because you can actually look for the guide RNA that is present.
It's a great update of a classic technique where
so
you
in
try
contrast,
to inhibit infection across
Yes.
the whole population, and the cells that you successfully inhibited infection in will survive. And so then you pull those out and say, hey, what inhibited the infection?
Now, how did we do it? How did Kathy Mendelson do it in the late 80s? We did it the opposite way. We asked, is there a gene from human cells that we put into mouse cells that can make the mouse cells make a polio receptor? Because we knew that mouse cells did not bind poliovirus, but we also knew that if you took polio RNA and put it into mouse cells, it would reproduce. So there is just a lack of a receptor inhibiting the ability of a poliovirus to infect mouse cells. So Kathy took DNA from human cells and sheared it because it's too big, and then added it to mouse cells, to many, many millions of mouse cells in culture. And then what do you do? So this was our puzzle. We said, okay, if we infect them, they're going to kill the cells that got the receptor gene from the human cells. So that's not a viable approach. And then it turned out that Eckhard Wimmer had acquired a monoclonal antibody that blocked poliovirus attachment to cells from a collaborator in Germany called Peter Nobis. And he gave it to Kathy. Kathy coupled that antibody. Again, the antibody is presumably against the receptor of the virus. We didn't actually know until we did the experiment. She coupled it to red blood cells, which are really big, and then she flooded the plates that she had added human DNA to mouse cells. So the mouse cells take up the human DNA, they make whatever proteins encoded in it. And if there's a poliovirus receptor on the surface, the monoclonal will bind to it, and you can look under a microscope and you'll see the red blood cells sitting on the plate. Such a crude, old-style
Yes.
approximation of what we can do now. But then she picked up those colonies with a pipette and purified them, and indeed some of them Now you can grow a lot of those cells and show, they can be infected with poliovirus. And then you have to pull out the receptor gene. We didn't have a guide RNA sequence to pull it out. We used a trick, which is so clever, but it's old school. Every 5,000 bases in human DNA, there is a repeat sequence called an ALU element, ALU. And this is a repeat. It's actually it's a transposon, and it's present many copies in the human genome. So basically, any human gene is likely to be next to an ALU sequence. So she got a clone and a piece of this ALU sequence in a plasmid and used it as a probe to identify. It was very, very complicated. And to her credit, she got it to work and identified the gene. And there's so many places where that could go wrong. And that's it. Now here.
Yeah, so that involved a tremendous amount of hard work, some very, very clever experimental design, and a healthy dose of luck. Because it turns out the antibody was against the poliovirus receptor single protein was sufficient to provide entry to the virus. And yeah, she managed to get that out. Really cool piece of old school science.
I mean, it could have turned out to be an allosteric inhibitor.
Yes.
A protein next to
of course, totally.
So
totally,
she
totally,
absolutely just fell into a big pot of gold.
We had a lot of faith in this experiment. And the thing is, it wasn't going to take her forever to get an answer. In theory, it was a couple of months that you could do this experiment. But she got colonies binding red blood cells the first time. I remember I was in France and she called me. She goes, "Oh my God, I got red blood cells." Oh my gosh. So sometimes you get lucky.
So Vincent, is there a way to inhibit the virus from being taken in by the cell once it attaches to the cell?
Yes.
Well, then why couldn't you then?
Well, we didn't. For poliovirus, we didn't know any way to do that.
Right.
And did you know what part of the poliovirus was binding to the?
No, we didn't. We had no idea.
Didn't know that either. Okay. Fine.
We didn't.
And
know
you know
anything.
everything now, right?
We know a lot, but boy, back then we didn't know very much, and it is just blind faith, right? That's a saying, blind faith, right? It also was a good band, which I really liked.
So in this, they approach this with a bit of faith too, but they're using a much more modern technique with the CRISPR-Cas9, and then you can do an actual positive selection here.
But you can buy the library, the CRISPR knockout library, right? For many cell types, they used HT29 cells, which is some colorectal or adenocarcinoma cell line from humans, right? And you buy it, you infect with the lentivirus, you let it cook for a while, and then you infect, and you see what's left. It's a week or two experiment at the most. And I don't mean to trivialize it.
No, because the initial, so this is one of those things where the initial experiment, I mean, as in Kathy's case, you get the initial experiment to work within a month or so. And then you got to figure out if the results you got is correct.
Yeah, so that's what they spend most of this paper showing, that what they pulled out, because they were very early on able to get cells resistant to parechovirus infection. So they used either A1 or A2, which both bind these integrins. They take the resistant cell populations and they look at the guide, they sequence the guide RNAs, and they identified two genes that are the most enriched in resistant cells, and that is the gene encoding the integrin subunit beta, which is part of the known receptor, and then MYADM, right? What is the name of it again?
Myeloid-associated differentiation marker.
Myeloid-associated.
MyADM is how I've been saying it in my head.
MyADM.
Is that, by the way, is that a receptor for something else in nature that?
Oh, yes, of course. It does not exist for parechoviruses, for sure.
No, of course not. But so that gets me back to my original thought about how clever, as you are used to saying, how clever viruses are. I guess it's like a safecracker that just keeps coming up with combinations until one of them clicks.
Yeah, in order to infect the cells, the virus had to find some protein that is on those cells, and so the selective pressure was to
I understand.
Latch onto what works, and this is
it. If you want to know what the function of MyADM is, it's involved in several processes, including negative regulation of heterotypic cell-cell adhesion, negative regulation of macromolecule metabolic process. Wow, that's a broad.
That's a lot of, yeah.
I'll say.
And negative regulation of protein kinase C.
That doesn't help me understand what it
Signaling.
does.
Yeah, it doesn't,
What
but
doesn't
it does
it
have,
do?
it has other functions, right?
Wow.
So how do you validate it?
Yeah,
Okay,
how
so
do you do that?
first you can, they knock the cell out in a couple of different cell lines that are known to be infectable with parechoviruses. You can knock them out with CRISPR, but this time you just have a guide RNA that matches this particular gene. You knock it out, and you try and infect with parechos, and they can't be infected if you knock out my atom. And if you put my atom back in, then they can be infected. So that's a classic experiment to show that a gene encoding a particular protein. And also they look at other picornaviruses, other enteros, like enteros A71 and Coxsackie B3. They do not care about my atom. They will infect those cells. So looks like it's essential for infection. That's all we know so far. It's essential for the virus to infect cells. They also do this experiment in intestinal organoids because these viruses are often found in stool. They're thought to be fecally, orally transmitted like poliovirus. So they're probably reproducing in the gut. We don't know for sure, but they're probably reproducing in the gut. So you can take stem cells and differentiate them into organoids, which kind of approximate the three-dimensional structure of the intestine, right? They have a basolateral and an apical domain. They have a lumen and so forth. And so they make these organoids either from wild-type stem cells or stem cells where they've knocked out my atom using CRISPR-Cas, right? And they show without my atom, the organoids can't be infected. With my atom, they can be infected. So in cell lines and in organoids, which approximate the gut, this is a protein is important for infection. They also do experiments to show what part of my atom is important. So my atom is a multipass membrane protein. So the protein threads the plasma membrane in and out multiple times. And so you get these loops of protein sequence on the outside and the inside. So they say probably these loops are important. When they compare the loops from different species that are known not to be infected with parechos, they say, oh, this seventh loop, is it the seventh? The last extracellular loop is divergent in amino acid sequence. So they do some swapping of that sequence with the human, and they show that this is, in fact, the part that's really important. They can even narrow it down to a couple of amino acids. So if the virus doesn't infect hamsters, it's because of some amino acid change in that 3 amino acids. I guess you could do the same thing with mice and make a mouse model, right?
Sure.
In theory. Now, this protein is essential for infection because that's the assay. We're putting virus in and we see that the virus is replicating. But is it, what part?
And
so
what part in it, yes.
Is it needed for viral RNA replication? And I would say, no, but you have to show it because it's on the plasma membrane. Why would it be needed for replication?
Who knows?
You never know. You never know. Right? You cannot assume things in science. If you do, you're going to be led down into a dark alley. And who knows what's going to happen there. So they take the viral genome, they cut out the region-encoded capsid proteins. So now this is a replicon. It's an RNA that you put in cells that's going to just replicate because it encodes the polymerase and it will copy itself. So you can take this RNA, which you can make, and you put it in cells, and you can measure its replication. They put a reporter, a luciferase reporter in it. So they take cells with or without my atom. Doesn't matter. It doesn't matter if it has my atom or not. The RNA still replicates. Okay, so you don't need my atom for RNA replication.
So if you bypass the whole entry process and put the RNA directly in the cell, it's fine. It doesn't need this receptor.
So that suggests that it might be an entry requirement.
Pretty strongly argues for this being an entry factor.
Yeah. Although there are a lot of things between entry. There's things between entry and RNA synthesis, yes. Right? But, okay, it turns out to be they're right, but... So the next thing they do does this bind the virus particle? That's a great experiment, right? Because if this is a receptor, it should bind to it.
Yep.
And so, we have 2 proteins that seem to be involved, maybe my atom and an integrin, right? So they do some attachment experiments, and they find that so you can bind virus to cells at 4 degrees, and then it will stay on the surface. And then when you raise the temperature to 37, the particles move into the cells by endocytosis.
Good times. Yeah.
You remember that, Alan?
Oh, yeah. Yeah, I did that. Many 10-minute time points.
Alan used to work on this
Yeah.
entry problem here for poliovirus. So if you knock out the integrin beta subunit, right, that's one of the ones they identified in their screen. We know that the virus is binding to integrins. If you knock out the integrin gene, you reduce binding of the virus particles, you reduce entry into the cell. And then if you put the integrin back, it restores all that. But if you knock out my atom, it reduces infection, but it doesn't really reduce binding all that much. So it's essential for entry, most likely, but not bind. The integrin seems to be the binder, and then somehow it's handed off to my atom, and that pulls it into the cell. And they do some other co-immunoprecipitation experiments that are basically consistent with that. If you you can see the entry of viral RNA into cells, even in cell. So if you mutate the gene encoding my atoms in that last loop, so that the virus won't bind, you impair the entry of viral RNA into cells, which you don't see that, of course, in cells with wild type receptor on the surface. So the data suggests that my atom is an essential entry receptor.
Yeah, they also do this low pH experiment, where they show that my atom does bind the particles at low pH.
That's right.
So it doesn't bind on the cell surface under neutral pH, but if you lower the pH, you can get it to bind to my atom. And again, it's if you mutate those residues to the whatever the horse residues, then it abolishes that binding. So it's consistent with a model. Again, you got to do more to figure out that this is really what's happening, but it fits with the idea that the virus probably binds the integrin at the cell surface, gets endocytosed into, one assumes, a lysosome that also contains my atom, pointing in. At low pH, when that acidifies, it binds my atom and uncoats, and they present this model in one of the figures. The paper's open access as well.
Right, so you need to do some experiments to figure out the exact sequence.
Sure.
But, and this is very different from poliovirus, where the virus binds the receptor right at the cell surface. It seems to make a channel in the particle, and that lets the RNA get in shortly after it gets into a vesicle at the cell surface. Yeah. So that's the story. It's quite interesting, and it illustrates how technology has changed in terms of identifying receptors. Now this will be used for many years because it's quite nice. Yeah. And as long as you have some assay, you can identify genes that are involved in it. And virus infection is one such assay that's easy to do. Now you're aware of parechoviruses, which is good. And
formerly known as echoviruses.
There you go. So I went to Columbia in 1982 wanting to identify the cell receptor for poliovirus. And I remember when I was a postdoc, my last month, I said to David, I think this would be an interesting project. And he said, no, probably not. So I said, well, then I'm going to do it because he won't work on it. And I get to Columbia, and in the fall, I present to the new graduate students what I wanted to do. And I left, and I went to my office, and Kathy Mendelson came running after me. She said, I want to do that. I want to do that receptor thing. It's okay. Come.
There you go.
And she did it. It was a really good job. And Kathy now is a professor at Columbia doing much better work than I ever did. So that's what you want. And you want people to do great things. All right. So that's, by the way, now one of the next things I would do is try and see if you can get a structure of the virus particle before and after this interaction that leads to release of the RNA. That would be interesting.
And that's another area where the tools have gotten a lot better in the past several years.
So, by the way, in case you're curious, if you infect 100 million cells with this lentivirus CRISPR-Cas knockout library, you will get 1,000 times coverage of all the open reading frames. 100 million cells. So a 10 centimeter plate, how many, is it 1 million cells on it or 10 million? Because that 6 centimeter had 1 million, I think.
I think it's 10 million on it.
10 million.
It's been a long time.
So 10 million in a 10 cent. Well, you could do a maxi plate too.
Yeah,
which would have
yeah.
many. And so this is not a lot of plates, basically,
Right.
that you need to do. And you can. So the way I would do it, I don't know how they did it, but you do the infection, and then you let the cells grow, and then I would trypsinize them to disperse them into single cells, split them into 2 plates, and then keep one and infect the other plate with virus. You keep one in case something goes wrong, right? Because that's your library. And then you infect the other cells, and you see what cells are left, and then you amplify those. That's the way I would do it. Memories. Does it bring back memories, Dixon? No.
He's muted.
Memories of what?
I'm just trying to engage you, that's all.
In my field,
Nobody does receptors, right?
no, we had different things to remember by, grinding up rats and
Rats,
isolating
the
an
Ratomatic.
RV
I
and
heard all about the Ratomatic from one of
the
your
Ratomatic, and that's
technicians.
right.
Yeah.
Only
now
eat the control rats. Do not eat these rats.
The next paper I picked because it's very cool. This one was cool too, but this one I think gives us some insight into severe influenza, but also maybe how to get around it. So this is a Nature article. Necroptosis, we'll explain that. Necroptosis blockade prevents lung injury in severe influenza. Many authors here. The first 2 co-authors are Gautam and Boyd, and the senior authors are Cooney, Thomas, Degterev, and Balachandran. They come from various places.
Fox Chase Cancer Center in Philadelphia, St. Jude Children's Research Hospital in Memphis, UC Santa Cruz, University of Houston, Tufts School of Medicine in Boston, University of Freiburg in Germany, and then a couple of companies, Reaction Biology in Malvern, Pennsylvania, and Vayu Therapeutics in Rancho Santa Fe, California.
So the pitch here is that can we find other points where we can inhibit influenza virus disease? And because that's useful, right? We only have a couple of antivirals. And they're saying that necrotic cell death is one potential point. So what is that? Necroptosis. It's a great word, right?
Yeah.
But
we used to think that necrosis was just kind of a
yeah.
default process that wasn't really regulated, just the cell kind of fell apart, whereas apoptosis was regulated cell death.
Right.
And
so
now we know differently.
These are different kinds of cell death. There's apoptosis, as Alan said, necroptosis, and then there's pyroptosis. They have different features, and they're regulated.
All of them are regulated,
ways.
yeah.
Yeah, and the point here is that influenza virus can kill cells, but the cells also initiate self-destructive programs. These are part of normal development when you need to get rid of cell populations, but they also are in response to pathogen insult, where the host recognizes that it's infected and says, sorry, cell, we're going to get rid of you to try and preserve the organism. And so this can cause a lot of damage. And so they tell us that this necroptosis accounts for most of the programmed death in influenza lung epithelial cells. All right. So this seems to be really a big part of the damage that's caused in the lung. And they say when this damage is unchecked, you get lung injury, you get severe illness, even without when the virus is cleared by your immune response, it can still have lung damage because of these necrotic processes, necroptosis going on. So they say, let's see if we can get an inhibitor.
And I gather, correct me if I have this wrong, but I think apoptosis is more of a surgical strike. One cell goes through an ordered disassembly, often by instruction from the immune system. Necroptosis is more like the nuclear option, and it's more likely to cause these adjacent sequelae. Is that
right?
Okay.
Yep. Yeah, and there's a good review article here I found, which is called Necroptosis, Pyroptosis, and Apoptosis, an intricate game of cell death. Actually, so this pronunciation has always been a bone of contention, whether
Yes.
to pronounce the P or not.
Yes.
And so you heard me saying apoptosis because
And I have been saying apoptosis. But
because I was excoriated once at a textbook meeting for saying apoptosis. Well, I said, isn't it helicopter?
right.
But they said, no, it's not. It's actually
It's helicoder. Right.
But a pyrotosis, that's weird. Pyroptosis. It should be pyrotosis and necrotosis, right? So I don't know. I'm just a virologist. I don't make up the words. Anyway, this review article talks about the differences between the three of them, and they're regulated. That's the point. We used to think necroptosis, as Alan said, was just necrosis. It was unregulated, a big knife and so forth, but it is regulated. All right. So, how do you get necroptosis? There's a host sensor protein called ZBP1, Dixon. You're going to have to remember these. There's going to be a test.
I'm sorry, I'm going to be absent that day.
There's a ZBP1 which detects zRNA, Z-form RNA made by influenza virus, and then activates a protein kinase called RIPK3.
RIP K3, get it?
Yeah, get it?
R.I.P. Rest
So,
in
in fact,
peace.
it actually stands for something, but the RIP is done on purpose.
Yeah, but
right?
exactly. Because
That's
what
right.
what it stands for is
Receptor
receptor interacting protein kinase 3, which is a completely non-descriptive name.
That's a
but they did it
stretch. That is
just
a
for the
big
abbreviation,
stretch.
so it's RIP, because it induces apoptosis and necrotosis.
All right.
This is from the same scientist that brought you hedgehog.
Yes.
So our ZBP1 activates RIPK3, which then phosphorylates MLK1.
Martin
It's
Luther
mixed.
King?
It's not mixed. It's not
MLKL.
MLKL. Sorry, it is not major league kinase. It's mixed lineage kinase like protein L, and that induces necroptosis. Okay, so let's review.
Please.
ZBP1.
Yep.
RIPK3.
RIPK3.
Milkle. Let's
Right.
call it milkle.
It
And then
phosphorylates milkle, and that leads to
Necroptosis.
necroptosis.
Then with necroptosis, you get pulmonary tissue necrosis. You get neutrophil recruitment. Neutrophils cause a lot of tissue damage
Yes.
and lung inflammation
Yes.
during severe flu, and that's really a big part of the problem. But you don't need it. You don't need necroptosis for CD8 T cell responses or for clearance of virus. Okay.
So
There's no
RIPK3
cytokine storm here.
Yeah, this inflammation is part of it. Yeah.
Oh,
So-
okay.
RIPK3 also can induce CASP8 protein activity and cause apoptosis,
Right.
but that does not require phosphorylation of CASP8. So in one case, you've got the kinase activity is required to lead down the path to necrosis or necroptosis. Without the kinase activity, you can still cause apoptosis, and that really is the crux of what they're going to try and do here.
So the cool thing is about what Alan just said is that, RIPK3 activates apoptosis, which can reduce viral loads because it's killing virus-infected cells. And you don't need necroptosis to do that. Because necroptosis, getting rid of necroptosis doesn't seem to play a role necroptosis doesn't play a role in clearance of virus, so apoptosis does. So they say, could we target just necroptosis with a drug and ameliorate the tissue damage and not worry about screwing up viral clearance, right?
Yeah, because you have the option of having either an effective clearance with apoptosis only or primarily, or you can have bad disease involving necroptosis. And for some reason, in some people, in some flu infections, you end up going toward necroptosis, and other people who are luckier or, caught a different strain or something end up having more apoptosis and they do better.
So they want to do some experiments to kind of proof of principle, right? So first, they have mice lacking MLKL, milkle, right? And those mice are going to be deficient in RIP3K-driven necroptosis, right? Because RIP3K, if you remember, Dixon, phosphorylates milkle, which is essential for the induction of necroptosis. So they have mice lacking milkle, which would be necroptosis specific, right? It's not going to mess with apoptosis.
Absolutely.
Very cool. So they take them and they infect them with a lethal dose of influenza virus, a PR8 strain. And they say, this should kind of give us an idea whether inhibition of necroptosis chemically would be useful at all. So all the wild-type mice die within 12 days. 70% of the milkle mice survive totally. It's not 100%, but it's pretty good, right? So maybe you can improve. Yeah. So they have no necroptosis, right? And they are being protected. 70% are protected. Now, in separate experiments that have been published before, if you take mice lacking ZBP1, remember that's the sensor, or RIPK3, they die because they can't induce apoptosis. So this is a nice proof saying that milkle is hitting necroptosis specifically. So if you can somehow inhibit necroptosis specifically, maybe we can make inroads here. Right?
We definitely don't want to inhibit apoptosis, because if you do that and stop necroptosis, then you die too.
Now, there are other RIPK molecules, right? There's 1, 2, and 3, and they show that if you inactivate one, the kinase activity of one, it doesn't protect the mice. So they have to go after 3. So they say we need a pharmacological agent that targets RIPK3 without interfering with RIPK3-induced apoptosis. And then that would be able to, in theory, protect the mice like these milkle knockout mice were protected. Okay.
Now, they're not the first ones to think of this.
No, they are not.
So, in fact, they are going to test this against 2 compounds that I assume were developed by GlaxoSmithKline, GSK843, GSK872. I think there actually might be a third. But these GSK compounds are the comparators, because I guess Glaxo tried to do this and their drugs ended up not working out so well. They had some on-target toxicities and other issues.
So it worked. It's an inhibitor of RIPK3.
It is an inhibitor of RIPK3.
But in vivo, or in cells, it didn't really have much activity for whatever reason. So they say, let's see if we can do better. And that's brave, folks, because this time it's not trivial to do better. It's not like the previous paper. Here you're going to have to make an investment. And they focus on a molecule that exists already. It's called PD180970. You don't have to memorize that, Dixon, because in a moment we're going to simplify it. It
doesn't matter. I just wrote it down.
It's a known inhibitor of RIPK2. That's not the one we want, but RIPK2 is related to RIPK3.
How many RIPs are there?
At least 3.
At least 3.
Well, at least 3. That's good.
Well, don't forget Riptide.
Yes,
that's right. Or Riptorn, the actor.
So they find that this PD molecule inhibits RIPK3, pretty good half maximal concentration, micromolar concentration. It was moderately potent at blocking necroptosis in cells. So moderately potent, not good enough, right? So what do? You call the chemists and you say,
make it better.
Make it better. So they generated 40 analogs, and one of them showed potent anti-necroptosis activity in cells. And that's a supplemental figure. Oh my gosh.
Yes.
Oh my God.
This is a modern Nature paper, which means that there's like an entire additional paper worth of supplemental data. By the way, it's also paywalled, so unfortunately,
that's right,
a lot of listeners are not going to be able to look at this. But there's just an enormous amount of work in this paper, and it becomes pretty obvious why, because they're aiming to produce a drug, which is a tall order.
So this new compound, Dixon, they named UH15-38.
Ask your doctor about UH15-38.
I would have named it that.
And this blocks this necroptosis in primary mouse fibroblasts. that were at concentrations that are lower than the GSK compounds that Alan mentioned. Okay. It prevents phosphorylation of milkle following stimulation. So you can artificially stimulate necroptosis in the absence of influenza virus by adding tumor necrosis factor. Okay. And they say this blocks phosphorylation of milkle in the presence of tumor necrosis factor. And
right.
it also
tumor necrosis factor, despite its name, and this has been a hard thing to get across to MDs especially, it is not a good thing in general.
Correct.
It's an inflammatory cytokine that is just bad news when it shows up.
Oh, you're right. So when I think about apoptosis, I think about the death of single cells. And when I think of necroptosis, do I think of death of a tissue rather than a single cell?
Necroptosis happens at the single cell level, but I gather it tends to have more sequelae in the tissue at large. That's why I was asking Vincent if that's a correct
Does it
perception.
aid in the spread of the virus in the tissue?
Necroptosis?
Yeah.
It doesn't inhibit it. Doesn't have any effect on virus clearance.
When a cell lyses, though, it's filled
Yeah,
with
you would
virus.
think, but it doesn't. You know, the whole animal studies show that if you knock out necroptosis, you don't affect virus clearance. It's apoptosis that plays a role in that. And I'm not sure why that would be. It's a good question.
Oh.
I'm not enough of an aficionado of cell death. It's supposed to be a funny there, but it's not really funny. All right, so they're thinking, all right, you know, UH15-38 must somehow inhibit RIPK3 differently from the GSK compounds. So what is it? They do some molecular modeling, and they find some substantial differences, basically, between the GSK and the UH. I've already forgotten the UH15, and in particular, it interacts with more parts of RIPK3 than GSK does, and they say maybe that's responsible for the increased potency. They look to see if it binds RIPK1 in vitro. It doesn't have any activity, and RIPK1 is involved in apoptosis, right? So that's good. There's no activity again. It's not going to inhibit apoptosis according to that. It's selective for RIPK3-driven necroptotic cell death. Okay, so let's now go to the mouse. They dose mice 30 milligrams per kilogram per day for 4 days, intraperitoneal. And lucky them, it accumulates in lung tissues. Oh my gosh. You know, that's exactly what you want. It's 8 times higher in lung than in plasma. So that's good,
it does accumulate
right?
in some other organs as well, but they're going to look at that later.
You know, the lung is where the influenza
Lung
virus.
is where you want it.
So then they give it to mice, caspase knockout mice, where the milkle has been tagged with an epitope so that you can track it with using antibodies. So in those mice, they lack caspase 8, and the reason that that's there is because it triggers multi-organ RIPK3 activation and phosphorylation of milkle, but the N-terminal epitope added to milkle prevents it from executing necroptosis. So what they can do is identify all the cells that have phosphorylated milkle because they have an epitope tag on it without them dying from a necroptosis, right? Because they want to see where this is working, but they can't if it dies from...
If it dies, yes.
...from necroptosis. So just by tagging milkle prevents it from inducing necroptosis. It's so clever. And I'm sure that was kind of accidental, right? Discovered at one point. Oh, we flag tag milkle. It doesn't work anymore, but someone
actually,
uses it.
That's good that it doesn't work.
It's good. So they find that, what does it mean? It means you can ask whether UH15 prevents phosphorylation of milkle. You don't have to worry about the cells dying from necroptosis. And they find that it prevents phosphorylation in every organ they test: lung, liver, heart, and kidney. So it probably could be used for treating diseases of other organs as well besides lung, right? I think it's a cool experiment that they use, that clever trick. How about safety? So they give a ton of, they actually first screen a bunch of protein targets, 50 critical protein targets. This is a typical thing that you do now in drug development. And these 50 targets are known to be associated with side effects in humans, right? We know this. And so they ask, will this interact with those 50 proteins? It doesn't seem to inhibit it. You have to run a lot of assays, activity assays for all these proteins. It doesn't. This is what contract research organizations are for, right? You give this to a CRO and you pay them a lot of money and they do the experiment for you. You don't have to burn through postdocs or graduate students. And that's a thing, right? So it doesn't inhibit any of these targets. So looks like it should be safe, but you never know until you test it. Then they give a lot to mice. There's no induction of apoptosis in any tissue. There's no general toxicity, so no liver toxicity, which you could measure by having liver enzymes enter the blood if you're killing liver cells. None of that. And they say it has a pharmacological profile that would be safe for deployment, in vivo deployment. So you could go into a clinical trial at some point. All right, then finally, what is the actual cell that's undergoing RIPK3-dependent necroptosis in lungs of mice infected with influenza virus? And they do single-cell analysis, sequencing analysis, and they say the type 1 alveolar epithelial cells are the prime replicative niche for influenza A virus in mouse lungs. And so they say, we're going to focus on these cells and ask whether our drug, UH15, can prevent necroptosis in those type 1 alveolar epithelial cells. So they isolate these cells from mouse lungs. This is another thing. You have to chop up the mouse lungs, make single cells. And then you use an antibody that will bind a protein specific for those alveolar epithelial cells. And then they couple those to magnets, and you can pull those cells out with magnets. It's called immunomagnetic selection. I just love it. And then they show that they're pure. They show that they undergo ZBP1-dependent cell death when you infect them with influenza A virus. If you take the same cells from ZBP null mice, they do not succumb to influenza virus death. They're mostly viable because they don't have the necroptosis inhibitor stimulator, right? And so they say, okay, what about UH15? Can it prevent influenza virus-induced necroptosis without blocking apoptosis? And the answer is yes.
Yes.
This drug blocks necroptosis in these, and again, this is type 1 alveolar epithelial cells cultured in vitro. And apoptosis still goes on, but no necroptosis. And so this drug, which blocks virus-induced phosphorylation of milk, is blocking necroptosis. And they also look at other influenza viruses and show that it works against a large panel of cells. So, and so clearly RIPK3 is important because that's what the target is of this inhibitor. All right, so then they do experiments in human cells to see if it works. They have to jigger human cells because most of them don't have one or more of the components of the necroptosis machinery. These are common cell lines, right, like HeLa cells. And so they jigger them to work, and then they can show that this drug at nanomolar concentrations protects them from influenza virus-induced necroptosis. So both in mouse and human cells, it works.
And they also use human lung sections. So one of the experiments, they took lung sections that had been surgically removed and looked at those and did a similar experiment and see that the UH15 also appears to inhibit necroptosis in those.
They say, this is a key point here, they say the drug exhibits wide separation of necroptosis inhibitory activity from the capacity to trigger on-target apoptosis.
Yeah.
Right? That's what you want.
So you can get it to inhibit apoptosis, but you've got to go to much, much, much higher doses than you use to inhibit necroptosis.
So, listeners, what do you think the next experiment would be? Dixon, what's the next experiment? You want to put this right in people now? No idea.
I'm an innocent here. I'm trying to follow this as best I can.
Well,
right,
no,
so
we've done this. So we've done this in cells. The next thing I think.
Well, you got to do it in vivo next.
So now we're going to look in mice. So we ask, can the drug prevent lethality driven by influenza
Okay,
virus?
and LD50.
Yeah.
So they infect wild-type mice with a lethal dose of influenza virus, and then they start treating them with the drug a day later, intraperitoneally. They give a range of doses from 7.5 mg per kilogram per day up to 50, and they look at survival, weight loss, and they compare them to mice with just the vehicle, no drug. Okay.
Right.
As low as 7.5 mg per kg per day prevents lethality in 40% of mice.
40.
And this is at a lethal, like way more than enough virus to kill all the
Sledgehammer
mice.
dose of
Yes.
virus.
And you can delay administration. You can do a shorter time course. You still prevent lethality in a significant number of mice, and you reduce the weight loss.
And if you do the experiment at a, I think it's an LD60 actually, but they knock the dosage down so that it's a little more realistic than the
Right.
way over the lethal dose. And at that point, anything above, actually any dose, all the doses, 1 mg per kg, you don't even have to go up to 3. All the mice survive, and the mice that got just the vehicle, just the solvent, 60% of them die.
That's right. So they call it a patient-relevant inoculum of
Yes.
influenza A virus. So it's fully 100% protective in mice when you give them that level.
And this thing with the delayed dosage was really interesting because they waited. They went anywhere from day 2 to day 5 before starting the drug regimen, and you can still significantly reduce lethality.
Yep.
out at when you give it at day 5, which is a big deal because a lot of people are not going to know they have the flu the day after they got infected.
Yep.
Now, they do the same experiment with one of the GSK drugs. No protection.
Doesn't go as well.
It's really, they have improved. Oh my gosh, it's just a great story, right?
Yeah.
They also check the H1N1 2009 pandemic influenza virus. It protects them. So both pandemic and seasonal strains it seems to work on. And it works even if you give it 5 days after infection, as Alan said. And they say this is an advantage over Tamiflu, which you have to give within 48 hours.
They're still in mice, and they're already trying to compete with marketed drugs. Yeah. Love it.
Right. But I mean, this is not going to reduce viral loads, right? But it's going to reduce the lower lung symptoms, which is lethality. That's good. So you're not going to feel better.
Right. Well,
But Tamiflu,
you will probably feel better than you would if you were
that's true.
getting necroptosis and being put on a respirator.
Oh, that's true. But I meant
Yeah.
like upper tract symptoms.
No,
are
you're not going to
be
suddenly, it's not going to be the Tamiflu effect of, wow, the symptoms resolved in a hurry.
or the Paxlovid effect with SARS-CoV-2. So the next thing they do, which is the last set of experiments, is to look at the lung itself to see. They've been looking at big reporters now, survival and weight loss. But if we look at the lungs, do we see an effect? So they do this same experiment. In fact, treat with UH15. Then they remove the lungs. And of course, they have a vehicle control. And the lungs from the vehicle treated mice, a lot of evidence of necroptosis at day 3 post-infection. By day 6, you see necroptosis in a big, substantial fraction of type 1 alveolar epithelial cells. The mice that get UH15, reduced numbers of necroptotic cells at both time points. And these are cells that are infected with influenza virus, right? So that the virus is driving the necroptosis. It didn't affect the number of cells undergoing apoptosis. You may say, how do you know this? Well, there are markers for necroptosis and apoptosis. You can stain the sections with antibodies against those, and you can tell. They also found that, so Dixon, you asked earlier about cytokine storm.
I did.
Right? So that is a thing. Inflammatory cytokines are released during infection of the lung, like IL-1 alpha, IL-33, IL-6, TNF, CXCL1. This drug, UH15, prevents the release of these from cells in culture, and they found the same thing in vivo. They can do bronchoalveolar lavage of mice, and they show that you get reduced levels of these cytokines when you treat with this drug. So this is a potential to dampening the cytokine storm as well. And finally, we have to look at the cells. So they take sections and look at, a pathologist examines them. So the lungs from infected mice that were treated with a UH15 show a decrease in alveolar damage and in the formation of hyaline membranes. They have reduced bronchiolar denudation and fibrosis. So they reduced, and they also reduce, the drug also reduces fibrotic lung damage, all of which are much higher in mice that get the vehicle. They have relatively normal lung function that they can measure. You can measure oxygen saturation, right? And airway resistance, and those are relatively normal. And then they can measure virus reproduction. They can do plaque assays to look for virus in the lung. The drug doesn't alter virus replication. It doesn't alter spread of the virus in the lungs.
It is not an antiviral.
It's not an antiviral. It does not negatively affect virus-specific CD8 T cells, which are important for restricting the virus infection. But it does dampen the influx of neutrophils. Remember, I told you at the beginning that neutrophil influx is a characteristic of necroptosis. And so those are reduced, and that reduces lung injury. And it does not affect virus clearance, which is carried out by CD8s in apoptosis. You couldn't ask for better results, really.
It's a beautiful paper about a very, very promising drug.
So what would you do with this? I mean, I guess you have people who are at risk for severe influenza. When they first start to show symptoms, you treat them, or do you preempt it and say, you're 85 years old, you have all these comorbidities, we're going to give this to you ahead of time if you test positive? I'm not sure what the clinical application is, but in theory, it can prevent pneumonia deaths, influenza pneumonia.
Yeah, give this when you give Tamiflu. And I don't know what the pharmacokinetics of this are. Is it orally bioavailable? Is this something that's going to have to be injected? So that could factor into it as well. I'm assuming they'd like to make it a pill, but I'm not a pharmacological chemist, so I can't. I don't know what the deal is here. I am sure that they're looking into all this.
I just searched the article for oral, and there's no mention of the word oracle. So, I mean, the mice, they don't do that.
They don't give pills to mice.
So that will have to be determined in studies in humans, I suppose, or something else.
Alan, that's because when you give it to them, they tell you that they took it, but they actually lied.
The mice lie, right? So that's what it means when you say mice lie.
That's right. They hid it underneath the straw.
So I hope we explained that to you clearly, folks. I want to make sure you understand this at a good level. And I know there are a lot of complicated abbreviations and terminologies, but it's both papers teach you a lot.
Yes.
About the science of virology. And now we will read some emails to see, in fact, how well you have learned other things that we have talked about. Dixon, can you take the first one, please?
I would be happy to. John writes, Vincent and Alan, And Dixon, too.
Yes.
I think particularly both of you were discussing claimed religious objections to vaccination in either 1107 or 1109. That brought to mind this one that crossed my window recently. Willpower dissolves in alcohol. Integrity dissolves in money, and reality dissolves in ideology. That might have relevance to the current campus protests that you commented on in 1109, too. I also liked your discussion about the mutability of viruses in the context of sloppy polymerases. It should often probably be mentioned more often than the nucleotide called at any position in the genome in a sequencing effort is the one that gives the strongest signal. But that doesn't mean that there weren't some strands in the mixture with one or the other 3 nucleotides at that position. Otherwise, unseasonably warm here in Greater Braddock, today at nearly 30 degrees C, as I imagine it was in New York and New Jersey as well. Cheers, John.
So the sequencing is an interesting point. It depends what method you use, right? If you use an old school method, yeah, you get an average of each base, and you cannot detect anything less than 15% of the total. But if you use modern high throughput technologies, right, the sensitivity is much greater, and you can see mixtures. You're always up against the error rate of the sequencing reaction, right? That has a certain error rate, so it's sometimes hard to distinguish that from the actual variation at any particular base. But when I sequenced polio back in 1980, it was a method that gave you one sequence only, even though we know now that that's not the case, right? Because it's an average, yeah.
Well, how many reads do you have to make to get it to be 100% accurate?
The more the better.
An infinite
The more
number.
the better.
To
The
get
more money, the
100%
better.
accuracy, yeah.
I did 2 reads.
2.
To both strands, yeah. One strand and one the other. 2 reads, 2x coverage, and now you can do hundreds, yeah. But there is an error in the sequencing, so that's why if you do many reads,
to make sure that you found the right area,
you can sometimes tell if an error is if it's
Gotcha.
only in one read, for example, then it's probably a sequencing error. But there are more sophisticated ways to do that. We should get people on who can talk about it. Alan, can you take the next one?
Sure. Hunter writes, TWiV personalities. I was pleasantly shocked when I read this article about animal sampling in the USA. Someone must have been listening to TWiV to come up with such a bold plan. It might seem foreign to many of the lab leakers, but it is refreshing to see that scientists are doing what scientists should do, try to answer questions. Hopefully, the project will give us some idea about the prevalence of SARS-CoV-2 in other animals besides humans. And provides a link that I have not read yet. And this is about a project. Oh, yeah. This is sampling 50 different animal species to look at how SARS-CoV-2 moves between people and wildlife.
Yeah.
These are wild
And
animals,
they're
right?
catching wild animals, bighorn sheep they've got photos of, and bunch of other things. I mean, as we've talked about, SARS-CoV-2 can infect many, many, many different species of mammals. So this is a very cool project to look at a whole bunch of different ones and see how it transmits.
Yeah, this is good.
Not how, like, mechanistically, but how, like, are the deer giving it to the raccoons? Are the skunks getting it from the mink? Are the, how is it passing through the population? What are the interactions there?
It's part of a program to monitor bighorn sheep health, and it was expanded.
Yeah.
That's very good. I like this.
Favorite quote from the article, she and Bowman, quote, feel really quite stupid at meetings when colleagues ask why the virus persists in deer, she says. Real scientists say things like this because it is true and they know when to say, we don't know. Take care and thanks for all you do. Hunter, who's a veterinarian, a retired food animal veterinarian. Cool.
Yeah, you have to say we don't know a lot, and then
Yep.
the non-scientists start to think that you're waffling or something. Volker writes, thank you for reading my email regarding the worldwide usage of IPV versus NOPV on epitope 1107. The two of you agreed to disagree, yet I kindly ask if you could reconsider. There are questions of taste, cheesecake versus strawberry cake, and of moral. What is more important, equality or freedom? Where there is no clear right or wrong and differing opinions are acceptable. Warning, rabbit hole alert. Then there are questions with definitive answers, laws of physics, Paxlovid versus ivermectin for COVID. These are amenable to the scientific method, and here disagreements should be resolvable. Here there are facts, maybe uncertainties, but after a thorough analysis, there is no room for opinions or alternative facts. Although we cannot perform large-scale repeated experiments on the IPV versus OPV question, I believe, for example, based on simulations, it falls into the second category. How about bringing a public health expert on the show to delve into the details? What would it take to use only IPV? What would this mean if we had to operate within the same financial resources? Please make sure both of you are available for this session. We are here. Thanks and best regards, Volker. P.S. The price of Paxlovid in Germany is 60 euros rather than $600 and even this small price is almost always covered by your insurance. It's tough to live in a country where the interest of big pharma is so little protected. All right. So Alan's argument was that we can't use IPV globally. We have to just accept the side effects of OPV. And I'm more idealistic, saying we should not have side effects and give everyone IPV. But admittedly, I do not know the logistics of injecting everyone with IPV. However, we did try and inject a lot of people with SARS-CoV-2 vaccines, right?
Yep.
We didn't get everyone for sure, but it seems like it's feasible. But I would still say I just don't like paralyzing kids.
No, I don't either.
Well, I know you don't.
Yeah, and that's why.
But that's driving me. That's what's driving me.
I completely see your point, and I could very easily take that side if I weighed things just a little bit differently. But as a pragmatist, the question, what would it mean if we had to operate within the same financial resources? Well, that's the current situation. So we cannot, with the current budgets given to public health, and that is an important distinction, we cannot deliver IPV to every kid in the world. With the current budgets given to the Defense Department, I'm pretty sure we could. Now, the question that would be interesting to have a public health policy person on about is where in between those, between the cost of an F-35 and the cost of that public health actually gets, would we have to be to actually get IPV out there? And I don't know what that number is. I do know that it's a lot more than what we have now. And so the choice is not IPV or OPV. It's OPV or nothing in poor countries.
Yeah, this is interesting.
I don't think nothing is the right choice.
I am not a pragmatist for sure.
I know. You're an idealist.
I'm an idealist, and I
like that about you.
I'm happy to be an idealist because sometimes idealists can influence pragmatists.
Yes, absolutely.
Doing things. I would like to get someone on who could speak to this.
Yeah.
I don't know who it would be.
It would have to be somebody in not just public health research, but public health connected with policy. There's got to be somebody at Columbia.
So, well, Steve Morse could certainly address it.
Paul Offit would have an opinion, I'm sure he would.
He would have an opinion.
We want someone who can say, no, this is why it can or cannot
Someone who's
happen.
actually sat at those tables of where people are budgeting for public health campaigns.
Well, if you have thoughts, folks, let us know. All right, we have two more left. Let's get to Dixon. Can you do the next one?
I can do the next one. Daniel writes, Dear TWiV team, the discussion on TWiV 1105 about the future of SARS-CoV-2 and whether it will end up like HCoV-OC43 from the 1889 pandemic, allegedly, reminded me of a study I discovered earlier in the pandemic originally from 2006 about OC43 outbreak in 2003. That's a lot of numbers. And then he gives a reference. An outbreak of human coronavirus, OC43 infection, and serological cross-reactivity with SARS coronavirus. In the summer of 2003, a respiratory outbreak was investigated in British Columbia, during which nucleic acid tests in serology unexpectedly indicated reactivity of severe acute respiratory syndrome coronavirus, SARS-CoV cases. Cases at a care facility were epidemiologically, he's quoting the article now, so I don't have a further wording on that. The study claims that OC43 can have a case fatality of 8% in long-term care settings. Is it possible that OC43 never fully lost its virulence and just faded into the background of ILIs, especially compared to influenza and RSV, especially without explicit testing? Would adapting the mRNA vaccines for OC43 spike make sense that could be given as a yearly booster for the elderly? Like, that's why you had me read that, I presume. Would Paxlovid work? Thank you.
So, Daniel, the virus is not the thing that's losing its virulence. It's population immunity that is resulting in a different pathogenicity pattern. And so in these long-term care settings, I'm not surprised that any virus might be more pathogenic than in the general population, because these are people with poor immune systems and other diseases. So, yeah, I think it's possible that OC43 could cause some severe illness. But overall, in the overall population, it's a very mild infection. And I think that's because it has gone through and infected. Most of the people have been infected at a young age, where they can make good memory B and T cells that last their lifetimes.
It also sounds to me like 8% is not a very high number for that age group. And especially in long-term care settings, there must be some underlying comorbidities that contributed to their fatality.
Exactly. There was one more that you skipped. Can you do that, Alan?
Sure. Robert writes, "Thank you for your informative podcasts that are great continuing education in virology and immunology. Interesting story of H5N1 in our milk supply, at least since February."
Yeah, this is a cool article, yeah.
A doctor in Sitka, Alaska, who graduated from Columbia PNS in 1972.
Look at that.
This is from Successful Farming.
Yeah.
Yeah, so this is an interesting story of how they figured out what was going on with the cows in Texas. Back in February, they were producing less milk. And so this story tells you how they found it was H5N1.
I'll be darned. And in case you're wondering where Sitka, Alaska is, and what the word Sitka might mean, it's actually the name of a very remarkable gymnosperm, or a pine tree. And it's they're remarkably beautiful and becoming more rare.
Neat. Okay, now it's time for some picks of the week, and we will start with you, Dixon.
Well, okie dokie. Everybody wonders whether there's life in other universes or other parts of our own galaxy. And, you tend to poo-poo the idea because, it takes a lot of stuff to come together to create the right conditions for life, period. And we have never been able to do that, but we think we were the mixing vessel where all those things came together in a remarkable fashion. But here's a new look, because now we have the James Webb Space Telescope, and with a much higher refinement for the spectroscopy that you can use to say what's out there in terms of molecules. And they actually looked at a nucleus of star formation, where the dust clouds are coming together and condensing and forming stars. And in that cloud that they looked at, they also looked at the complex carbon compounds that you could identify by mass spectroscopy. And it is quite remarkable to see how many different familiar molecules, but more than 3 or 4 carbon lengths in length, were identified by the Space Telescope. Now, carbons, they come together, but they also come apart because there's a lot of radiation out there, especially when a star ignites and forms its first radiation release, so to speak. But in addition to that, of course, in his star clouds, you can get planets forming. And so the the way you get a planet with the chances for life is to include those molecules right away so that they have a head start. And this, I was just blown away by this. I must say that when I saw this, first I passed over it and then I came back to it and I said, wow, now we have a way of looking to see whether or not we can identify concentrations of carbon-based molecules that would go even further than that. We found amino acids. I don't know if we found nucleic acids or not yet, but we're certainly looking intensely for them. But now it's really an amazing thing to know that we can use something as remarkable as a space telescope to serve the universe and see what we find.
This is really cool.
It's
Yeah,
amazing.
I think it is, actually.
Yeah. And they
Yep.
found formic acid, so there are
It
ants in
did.
that.
Ants.
There are ants in that nebula.
Yeah, they're ants.
Exactly. Exactly.
Or am I leaping to conclusions? Maybe a little bit.
All right, ants. And then there's ethanol somewhere there,
Yeah.
and then there
But
must
of
be bars,
course.
right? They're
Yeah.
bars. Very cool. All right, thank you, Dixon.
You're welcome.
Alan, what do you have for us?
I have a calculator that I
Oh.
found a little while ago. So I was planning a trip. I'm actually going to be at ASV briefly. I won't be at the TWiV, but I will be giving a presentation in one of the workshops. And I was planning my trip and going out to Ohio, and gee, how do I get there? Well, you can't take the train because the Amtrak doesn't go to Columbus. So I'm going to fly, or does it make more sense to drive? And gee, how would I even figure that out? And it turns out there are a bunch of these climate calculators online. This one, as far as I can tell, it's the most reliable looking one that I found. And I cross-checked it against a few other sources. What it allows you to do is put in, it's got a bunch of different tools on it, but what I used is the flight and the car tool. And you can put in your trip. Where are you going from and to? The one drawback is it's all in Canadian units, so you have to convert to kilometers and liters of fuel per 200 kilometers instead of miles per gallon. There's a little bit of additional work you need to do. But the cool thing with the flight calculator is you can tell it your exact route. So if I would be taking Southwest from Hartford to BWI to Columbus. And they always fly 737s. You put in the type of plane, and it takes all of that into account, and it's drawing its numbers directly from the International Aviation Organization's database on fuel consumption for aircraft and the effects of that fuel expenditure at altitude, which is a significant thing with aviation. I calculated that it's actually a wash. I could fly or drive for that particular trip, and my carbon footprint will be equivalent. It would be different if I were going to be sharing the car with somebody. Then it would make more sense to drive.
So the answer is rent a Tesla.
Yes. That just shifts the carbon footprint.
That's right. Well,
Ah.
the shoe is on the other foot. That is
Right.
exactly right.
I was just, it just flew to Columbus. It's just an hour
Okay.
each way.
Huh.
But yeah, driving is longer. So I can
Or
see
you
Driving
what it would be.
it's a 10-hour drive from where I am, and I was not looking forward to it, but I thought, there's a big difference in the footprint. I might try and plan a multi-day trip and
So what are you
camp
going
or
to do?
whatever.
Fly?
Just
I'm going to
out
fly.
of curiosity, Alan, what would it cost to rent an airplane? A little Piper Cub or a Comanche or something like that?
Yeah, so I don't do that anymore, but that would be, it's more weather dependent because you're flying
Well, of
down
course.
in the weather. But it would be about $150 an hour for the planes I used to fly, and hours to Columbus. It would probably be about a 4-hour trip each way. We're over $1,000.
Right.
No, you can fly for less.
Yeah, you can. Yeah, I mean, it's since airline deregulation, it has not been cost effective to fly your own airplane, and it is definitely not carbon effective. The carbon footprint for that trip would be far worse than either the car or the airline.
All right, thank you. My pick is a pick I made in January, but I want to do it again because it's my lectures in virology, my virology course at Columbia University, which just ended. And now all 25 lectures are on YouTube. So you can go watch them. And I'm sure some people don't know this. So I want to pick it again. So again, from the beginning, it's all about how viruses reproduce in cells. And then at the end, how they cause disease, how we attempt to prevent disease, how we vaccinate, how viruses emerge, evolve, etc. And then the last lecture is about therapeutic viruses, modifying these viruses so they can help us. So it's a complete introductory virology course. It is free. You can watch the lectures and learn something about viruses.
Nice.
So check that out. We have a listener pick from Alan, who is not Alan Dove.
Not me.
Greetings from the editorial offices of the Journal of Clinical Ambivalence. Here's an NPR story and interactive quiz on pandemic respiratory disease transmission based on the new WHO guidelines. The staff here got 100%, mostly thanks to careful TWiV listening. Now, wait a minute. Is there a journal called Clinical Ambivalence?
There should be if there isn't.
No, that's a joke.
The Journal of Irreproducible
Yeah,
Results.
so this is when I saw this in my news feed, I immediately went and took it, and I also got 100% on it.
You got 100%?
Yeah, which I was happy to see that.
There's no link here. Oh, here
I
it
just
is.
included it.
Did you put it in? Thank you, because
Yes.
when I copied it, it didn't go over. All right, very good. Thank you very much. And then from AZ, a short video by Dr. Rob Swanda explains why has H5N1 bird flu infected other mammals more readily. Humans lack one type of cell receptor in the upper tract that H5N1 viruses can use to establish an infection. So let's see. H5N1. Who is Rob Swanda? I don't know, but I didn't look at this, but we will... let's see, 1.6 thousand views. That's a good sign. Because if it was fake, it would have 1.6 million views. What do you think of that metric, huh?
He is says he's a PhD, Dr. Rob Swanda, making science accessible. This sounds good. We probably ought to meet this guy.
He has a website.
Medicine for thyroid disease.
He's a science
very into thyroid disease, it seems.
Okay.
But he's got a bunch of other stuff.
Anyway, I don't know what he is saying is why bird flu has infected other mammals more readily. If there has been a receptor change or what. I should know this, right? Do you know offhand, Alan, if there's a HA change to include alpha-2,6-sialic acids?
I don't know. I'm still trying to figure out who Rob Swanda is. Yeah, he's mostly about thyroid dysfunction.
Yeah, so they combine alpha-2,6-sialic acids, and that may in part explain why they're infecting many more mammals. So, but I will watch this. Thank you, AZ. That'll do it for TWiV 1111. 1111. You can find the show notes at microbe.tv/TWiV. You can send us questions, comments, picks of the week to TWiV at microbe.tv. And we'd love your financial support so we can continue to do these programs. You can go to microbe.tv/contribute for various ways where you can do that. Dixon Despommier can be found at trichinella.org and thelivingriver.org. Thank you, Dixon.
You're quite welcome. What a joy it is to be a part of this group.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at Virology. No, not anymore. Well, old columns are at virology.ws, but I haven't written in a long time. You can find me at microbe.tv or here at the incubator. Come visit. Send us an email. We'd love to have you. Some students are going to visit next week from my class.
Cool.
Oh, great.
I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.